Tervetuloa Lassi Heinilän väitöstilaisuuteen. Vastaväittäjänä toimii professori Nicolas Inguimbert Perpignan yliopistosta Ranskasta. Welcome to public examination of PhD thesis by Lassi Heinilä. The opponent is Professor Nicolas Inguimbert, University of Perpignan, France. Tiedekunnan määräämänä kustoksena julistan väitöstilaisuuden alkaneeksi. As the custos appointed by the Faculty of Agriculture and Forestry, I declare this public examination open.
Okay. Dear custos, dear opponent, ladies and gentlemen, I will start this defense with the lectio precursoria, which is a brief summary of my thesis aimed for the general audience. I will give this presentation in Finnish with the slides in English. Tutkin työssäni cyanobakteerien eli sinilevien tuottamia luonnonyhdisteitä. Cyanobakteerit kehittyivät noin 2,5 miljardia vuotta sitten. Cyanobakteerit olivat ensimmäisiä organismeja, joille kehittyi kyky happea tuottavaan yhteyttämiseen. Tämä yhteyttämisreaktio oli tehokas energian ja hiilen lähde, ja cyanobakteerit yleistyivät. Niiden lisääntyessä maapallon hapettomat meret ja ilmakehä muuttuivat hapellisiksi ja mahdollistivat happea energiantuotannossa käyttävien organismien synnyn. Cyanobakteereista kehittyi myös kasvisolujen viherhiukkasia endosymbioosin kautta. Seuraavien miljardien vuosien aikana maaneliöstä on muuttunut jatkuvasti ja myös cyanobakteereita ravintona käyttäviä ja kilpailevia eliöitä on syntynyt. Cyanobakteerit ovat samalla kehittyneet vastaamaan näihin ympäristön luomiin biologisiin uhkiin ja myös abioottisiin uhkiin, kuten säteilyyn. Tärkeä osa cyanobakteerien aineenvaihduntaa on erilaisten bioaktiivisten molekyylien tuottaminen, joilla ne vastaavat näihin uhkiin. Sinilevät tunnetaan yleisesti niiden muodostamista laajoista kukinnoista, jotka esiintyvät vesistöjen pinnalla laajoina lauttoina, lämpiminä ja aurinkoisena kesinä. Sinilevän kasvun rajoittavana tekijänä ympäristössä on yleensä fosfori, joka vesistöjen rehevöityessä mahdollistaa kukinnot. Kukinnot tuottavat monina ihmisille ja muille eläimille myrkyllisiä aineita ja estävät veden käytön talousvetenä ja virkistäytymisessä. Yleisimpiä myrkkyjä ovat makean veden bakteerien tuottama mikrokystiini ja murtovesien ja suolaisen veden bakteerien tuottama nodulariini. Nämä esimerkkiyhdisteet ovat kuitenkin vain pieni osa lukuisista yhdisteistä, joita sinilevät tuottavat. Tutkijat löytävät jatkuvasti uusia yhdisteryhmiä ja tunnettujen yhdisteiden variantteja. Näitä yhdisteitä tutkitaan niiden mahdollisten lääketieteellisten sovelluksien vuoksi. Monet yhdisteet esimerkiksi estävät toisten mikrobien, virusten tai syöpäsolujen kasvua. Tällä hetkellä markkinoilla on yksi cyanobakteerien tuottamista yhdisteistä johdettu lääke, dolastatiini yhdisteryhmästä johdettu brentuximab vedotin, jolla hoidetaan imusolmukesyöpiä. Lääkekehityksessä yksi tapa lähteä liikkeelle on johtolankamolekyylien etsiminen. Tällaisia molekyylejä ovat monenlaiset luonnonyhdisteet. Uusia bioaktiivisia molekyylejä löydetään jatkuvasti. Nämä johtolankamolekyylit sopivat kuitenkin harvoin sinällään lääkeaineeksi. Ongelmaksi voi muodostua esimerkiksi yhdisteen yleinen myrkyllisyys, huono liukoisuus tai epäspesifi aktiivisuus. Näiden yhdisteiden sisältämiä rakennemuunnelmia voidaan kuitenkin kokeilla osana toista yhdistettä lääkeaineen optimoimiseksi, tai niihin voidaan lisätä uusia elementtejä niiden kuljettamiseksi kehossa oikeaan kohteeseen. Yhdisteiden tutkimisessa oleellista on yhdisteen rakenteen vaikutus sen bioaktiivisuuteen. Se, millaiset variaatiot tekevät yhdisteestä tehokkaamman tai spesifimmän. Tämän lisäksi on hyödyllistä selvittää, miten yhdiste tuotetaan solussa. Tämä perimän DNA-tieto on kallisarvoista. Sitä voidaan käyttää rakennettaessa geneettisesti muokattuja solutehtaita lääkeaineiden valmistamiseksi. Itse asiassa suuri osa tällä hetkellä markkinoilla olevista lääkeaineista tuotetaan siirtogeenisissä mikrobeissa. DNA-sekvenssitietoa voidaan käyttää myös, kun etsitään uusia luonnonyhdisteitä. Vertaamalla tunnettuja biosynteesigeenejä ja toisten bakteerien sekvensoituihin genomeihin voidaan löytää aiemmin havaitsemattomia biosynteettisiä geeniryppäitä. Suuri osa luonnonyhdisteistä tuotetaan solussa niin sanotuilla ei-ribosomaalisilla proteaanisyntetaaseilla, lyhennettynä NRPS-entsyymeillä sekä polyketidisyntaaseilla, PKS-entsyymeillä. Nämä ovat suuria modulaarisia megaentsyymejä. NRPS-entsyymit koostuvat moduuleista, ja jokainen moduuli tunnistaa ja liittää yhden aminohapon kasvavaan peptidiketjuun. Moduulin alayksikköjä ovat katalyyttiset domeenit, joita edustavat nämä pallot täällä kuvassa, joilla on omat tehtävänsä substraatin käsittelyssä. PKS-entsyymit ovat järjestäytyneet samalla tavalla moduuleiksi ja niiden alayksiköiksi domeeneiksi. PKS-entsyymien raaka-aineena substraattina toimivat aminohappojen sijaan asuliryhmät. Täällä näin. Kuten kuvassa asetyyli. NRPS-entsyymeille tyypillistä ovat monenlaiset aminohappojen muokkaukset osana prosessia ja epätavallisten aminohappojen lisääminen yhdisteeseen. Cyanobakterien tuottamille yhdisteille yleistä on myös yhdistynyt hybridi PKS-NRPS-tuotanto. PKS-NRPS-hybridi biosynteesireiteillä voidaan rakentaa yhdisteitä, joiden rakenteessa on sekä aminohappoja että polyketidiketjuja, kuten rasvahappoja. Rasvahappojen hydrofobinen luonne vaikuttaa oleellisesti syntyneen lipopeptidin mahdolliseen vuorovaikutukseen muiden yhdisteiden ja solun osien kanssa, kuten solukalvon kanssa. Tämän työn tarkoituksena oli etsiä ja kuvata uusia bioaktiivisia yhdisteitä, joilla olisi lääkkeellistä potentiaalia. Työ oli perustutkimusta, jossa tähdättiin uusien rakenteiden löytämiseen ja niiden biosynteesireittien kuvaamiseen. Työn painopisteenä olivat sienten kasvua estävät yhdisteet, joita voitaisiin käyttää sieni-infektioiden hoitamisessa. Tässä käytettiin monenlaisia mikrobiologian, kemian ja bioinformatiikan menetelmiä. Yhdisteiden tutkimiseksi täytyi ensin kasvattaa suuri määrä niitä tuottavia bakteereja. Tuotettu biomassa kuivattiin ja uutettiin. Tutkittavat yhdisteet puhdistettiin uutteesta kiinteäfaasiuutolla ja nestekromatograafisesti. Puhdistettujen näytteiden kemiallinen rakenne selvitettiin massaspektrometrialla ja ydinmagneettisella resonanssispektroskopialla. Uutteiden ja puhdistettujen yhdisteiden bioaktiivisuutta testattiin agar-diffuusiomenetelmällä tai spektrofluorometrisesti. Tutkituista bakteereista eristettiin DNA, joka lähetettiin sekvensoitavaksi. DNA-sekvenssi analysoitiin bioinformatiivisilla ohjelmilla. Geenien tarkemman funktion selvittämiseen tehtiin manuaalista vertaamista tietokantoihin ja kirjallisuuteen. Biosynteesiin osallistuvien räätälöivien geenien toimintaa tutkittiin kokeellisesti siirtämällä niitä kolibakteeriin. Ensimmäinen tässä väitöskirjassa käsiteltävä vertaisarvioitu artikkeli kertoo skytosyklamidi-yhdisteiden tutkimisesta ja kuvaa skytosyklamidien biosynteesireitin. Skytosyklamidit kuuluvat tunnettuun yhdistöjoukkoon laksafysiineihin. Laksafysiinien rakenteita ja bioaktiivisuutta on tutkittu ennenkin, ja niistä on löydetty toistakymmentä erilaista varianttia. Laksofysiininen biosynteesireitti on tähän asti ollut tuntematon, ja kuvaan sen tässä artikkelissa ensimmäistä kertaa. Tässä kasvatin Skytonema hoffmannia PCC 7110 bakteerikantaa. Kantaa oli ryhmässämme tutkittu aikaisemminkin ja todettu sen estävän sienten kasvua. Tuolloin sienten kasvua estävä yhdiste jäi tuntemattomaksi. Nyt bakteerikannan kemiallisessa analyysissä yhdisteiden huomattiin olevan laksofysiinivariantti skytosyklamideja. Skytosyklamidit A, B ja C on kuvattu aikaisemmin, mutta löysimme myös 3 uutta skytosyklamidivarianttia A2, B2 ja C2. Laksofysiinit jaetaan 2 ryhmään. 11 aminohappotähteen A-tyyppiin ja 12 aminohappotähteen B-tyyppiin. Näiden 2 tyypin välillä on useissa tutkimuksissa kuvattu vahva muiden eliöiden kasvua estävä vuorovaikutus, yhteisvaikutus jopa. Kuvassa näette skytosyklamidien rakenteen. Tässä vasemmalla 11 aminohappotähteen A-variantti ja oikealla 12 aminohappotähteen B-variantti ja C-variantti. Skytosyklamidit sisältävät monia epätavallisia aminohappoja. Beta-amino-oktaanihapon, hydrobuturiinin, hydroksiproliinin, homoseriinin, hydroksi-D-leusiinin, hydroksiasparagiinin, muita D-aminohappoja ja N-metyyli-isoleusiini. Tutkin puhdistettujen yhdisteiden sienten kasvua estävää vaikutusta agar-myelodiffuusio-menetelmällä, jossa kasvatusalustaan diffuntoitunut yhdiste estää sienten kasvun. Jättää mallille estokehän. Kuvassa näette mallilla Aspergillus flavus homekasvuston, jonka keskellä on pahvikiekko, johon on annosteltu skytosyklamidia. Kuvassa vasemmalla skytosyklamidi A2 ja oikealla B2, joiden ympärillä näkyy vain aavistus estokehästä. Laksofysiineille ominaista on näiden 11 ja 12 aminohappotähteen tyyppien välinen synergistinen yhteisvaikutus, joka nähdään 3. kuvassa, jossa kiekolle on annosteltu molempia yhdisteitä. Skytonema hoffmannin PCC 7110 kannan sekvensoitu genomi löytyen GBank-tietokannasta. Käyttäen Antismash-työkalua tunnistimme genomista mahdollisen skytosyklamideja tuottavan geeniryppään, joka on kuvattu kuvan ylälaidassa olevilla nuolilla. Ja nuolien alle on kuvattu nämä geenien tuottamien PKS ja NRPS-moduuli ja domeenijärjestys. Ja alla on vielä näiden tuottamat 11 ja 12 aminohapon skytosyklamidit. Geeniryppässä on selkeästi 2 ryhmää NRPS-geenejä, jotka vastaavat skytosyklamidien rakenteen peptidiketjuista. Ensimmäiseksi sinisellä A-tyypin NRPS-geenit ylhäällä, moduulit tässä keskellä ja rakenne alhaalla. Ja vihreällä tai turkoosilla B-tyypin skytosyklamidien biosynteesigeenit ja moduulit ja rakenne. Tarkastelemalla moduulien annulaatio-domeenien aktiivisia taskuja voimme ennustaa, minkä aminohapon moduuli liittää peptidiketjuun. Nämä ennusteet on kirjoittu moduulien alla ja vastaavat yhdisteessä olevia rakenteita. Esimerkiksi täällä fenyylialaniini, joka löytyy sitten meiltä täältä rakenteesta. Tyypillisesti molemmilla NRPS-tuotteilla olisi omat biosynteesin aloittavat PKS-moduulit, mutta tässä geeniryppässä onkin harautuva biosynteesireitti. NRPS-entsyymit jatkavat yhteisten PKS-entsyymien aloittamaan biosynteesiä harautuvalla reitillä, joka oletettavasti aloittaa biosynteesin kiinnittämällä oktaanihappoon, jonka biosynteesigeenit ovat myös täällä nämä PKS-geenit ja moduulit kuvattu ja oktaanihappo täällä ensimmäisenä rakenteessa. Toisessa artikkelissani kuvaan lisää laksofysiinivariantteja. Tässä projektissa seuloimme cyanobakteerien kantokokoelmaamme tarkoituksena löytää sienten kasvua estäviä yhdisteitä. Cyanobakteerikanta nostok UHCC 0702 osoittautui antifungaaliseksi. Tarkemmassa kemiallisessa analyysissä huomasimme myös tämän kannan tuottavan laksofysiinivariantteja, jotka ristimme heinämideiksi. Heinämidien rakenteessa on runsaasti ei-proteiinogeenisia aminohappoja. Osa näistä modifikaatioista ei ole ennen tavattu laksofysiineissä. Beta-aminohydroksioktaanihappo, hydroksimetyyliproliini ja O-karbamyylihomoseriini. Heinämidien bioaktiivisuutta tutkiessamme huomasimme saman yhteisvaikutuksen kuin skytosyklomideilla ja muilla laksofysiineilla. Kun sekä A- että B-variantteja yhdistetään, estävät ne sienen kasvua paljon tehokkaammin kuin yksittäiset yhdisteet. Eristimme DNAn heinämidien tuottajakannasta ja sekvensoimme genomin. Kokonaisesta genomista löysimme heinämidien biosynteettisen geeniryppään, joka on hyvin samanlainen kuin skytosyklamideilla. NRPS-entsyymien domeenien organisaatio on identtinen skytosyklamidien geeniryppään kanssa. Ryppäissä on kuitenkin eroja. Heinämidien ryppäästä näyttää puuttuvan koko prosessin aloittava FAL-domeeni. Tällainen domeeni kuitenkin löytyy genomista, tosin 1,6 megaMS:n päästä sekvenssistä. Tämä on epätavallista, mutta annotoimme geenin osaksi geenirypästä sen ollessa järkevin vaihtoehto biosynteesireitin toiminnalle. Tein ennusteen myös rakenteen sisältämään hydroksiproliinin ja hydroksimetyyliproliinin räätälöivistä entsyymeistä. Nämä geenit tai entsyymit nimeltään LXA, N, O, P, Q ja R. Nämäkin geenit sijaitsevat etäällä NRPS-geeneistä. LXA, O, P, Q ja R geenit ovat huomattavan samankaltaisia tunnettujen metyyliproliinia tuottavien entsyymien geenien kanssa, joka johti oletukseen niiden tuottamasta hydroksimetyyliproliinista. Tutkiessani näitä geenejä huomasin, että LXAN-geenille on homologi eli samankaltainen geeni myös kytosyklamidia tuottavan geeniryppään läheisyydessä. Tämä geeni voisi olla molemmissa rakenteissa esiintyvän hydroksiproliinin taustalla. Kloonasimme geenin kolibakteeriin, jonka jälkeen huomasimme bakteerista löytyvän hydroksiproliinia. Yritimme kloonausta myös oletetuilla hydroksimetyyliproliineja tuottavilla geeneillä onnistumatta havaitsemaan tuotetta tai sen välimuotoja. Kolmannessa artikkelissa tutkin brasilialaisen Nostoc Sena 543 bakteerin tuottamia yhdisteitä. Osana laajempaa brasilialaisten kantojen seulontaprojektia tämä bakteeri osoittautui mielenkiintoiseksi kemiallisten ominaisuuksiensa perusteella. Kanta näytti tuottavan tuntematonta aeruginosiinivarianttia ja nodulariini maksumyrkkyä. Kantaa tutkiessamme kuvasimme uuden yhdisteryhmän, jonka ristimme pseudospumingiineiksi sekä kuvasimme niiden biosynteesireitin. Osoitimme yhdisteiden estävän trypsiiniproteasien toimintaa toimivan trypsiinin kaltaisten entsyymin toiminnan estäänä eli inhibiittorina. Pseudospumingiinit ovat tetrapeptidejä, jotka muistuttavat cyanobakterian tuottamia aeruginosiineja ja spumingiineja. Tämä variantti pseudospumingiini A koostuu 4 alayksiköstä: hydroksifenyylimaitohaposta, homotyrosiinista, isoleunisiinista ja argininaavista. toisessa ja kolmannessa hapossa on variaatiota. Paikoilla voi olla homofenyylialaniini, leusiini tai valiini. Pseudospumikiinit eroavat spumikiineistä, sillä spumikiineille tyypillistä proliinia tai metylproliinia ei ole, ja aerogenosiineista, sillä aerogenosiineille tyypillistä joy-aminohappoa ei ole. Eristin nosto Xeno 543 kannan DNA, josta sekvensoitiin kokonainen genomi. Genomista löysimme spumikiinin geenirypästä läheisesti muistuttavan geeniryppään, jonka päättelimme tuottavan pseudospumikiinia. Ryppäät ovat PKS, NRPS, domeenijärjestykseltään identtiset, mutta ero löytyy metyyliproliinia muokkaavista geeneistä. Spumikiini ryppäästä geenit nimittäin löytyvät täältä SPU, C, D, E. Mutta pseudospumikiinin geeniryppäässä niitä ei ole. Tämä ilmeinen ero johtaa eroaviin tuotteisiin ja kolmannen aminohapon tunnistavan adenolaatiodomeenin substraatti-spesifisyyden eroon, toisen tunnistaessa haarketjuisia aminohappoja ja toisen tunnistaessa metyyliproliini. Pseudospumikiinin bioaktiivisuutta tutkittiin spektrofluorometrillä. Kokeessa testattiin, miten pseudospumikiini estää trypsiini-entsyymin toimintaa. Koeliuokseen laitettiin tarvittavat reagenssit ja pseudospumikiini. Kokeessa seurattiin trypsiinin toiminnan vaikutuksesta syntyvää fluoresenssia. Jos pseudospumikiini esti trypsiinin toiminnan, ei fluoresenssia syntynyt. Kuvaaja näyttää tulokset useille eri pseudospumikiinien konsentraatioille. Kun pseudospumikiinia ei ollut liuoksessa lainkaan, kasvoi fluoresenssi vapaasti. Täällä nämä harmaat kolmiot. Ja kun pseudospumikiinia oli tarpeeksi, ei fluoresenssia päässyt juuri syntymään. Täällä nämä harmaat neliöt. Tuolla 80 noin mikromoolin konsentraatiolla. Lopulliseksi aktiivisuutta kuvaavaksi IC50-arvoksi laskettiin 4,5 mikromoolin konsentraatio. Joidenkin tunnettujen aerogenosiinivarianttien vastaava arvo on 50 nanomolaarinen konsentraatio, joka tarkoittaa noin 100 kertaa tehokkaampaa inhibitiota. Löysimme kannasta myös Suomenkin vesistöissä yleistä maksamyrkky nodulariinia. Nodulariinin konsentraatioksi määritettiin 4,3 milligrammaa grammassa kuivattua biomassaa. Tämä pitoisuus vastaa tunnettuja Itämeren kukintojen pitoisuuksia. Tulos on yllättävä, sillä muista kuin nodulaarian suvun bakteereista ei ole ennen löydetty vastaavia pitoisuuksia nodulariinia. Yhteenvetona työni tuloksista. Löysin ja kuvasin uusia laksafysiinivariantteja, skytosyklomideja ja heinamidit. Heinamideistä löytyi uusia rakenteita, A-karbamyyli, homoseriini ja hydroksymetylproliini. Kuvasin ensimmäisen ja toisen tunnetun laksafysiinian biosynteesireitin ja tein ennusteet joidenkin modifikaatioiden alkuperästä. Kuvasin kokeellisesti L-laksaa N-proliini hydroksylaasin toiminnan. Kuvasin myös pseudospumikiinien rakenteen, biosynteesireitin ja trypsiini-inhibitioaktiivisuuden. Määritin korkean nodulariinipitoisuuden odottamattomasta nostokannasta. Päätelmät ja johtopäätökset työstä. Laksafysiinit tuotetaan epätavallisella haarautuvalla biosynteesireitillä. Päätelmänä tästä niiden tuotantoa säädellään samanaikaisesti yhteisvaikutuksen aikaansaamiseksi. Kuvasin kemiallisia uusia rakenteita, joiden tuottamaa tietoa voidaan käyttää uusien luonnonyhteiden rakennetta tutkittaessa ja lääkekehityksessä. Laksifysiinien ja pseudospumikiinien biosynteesireitit ja rakenteet ovat uusia esimerkkejä PKS-NRPS biosynteesireittien geneettisten elementtien liikkumisesta ja niiden vaikutuksesta uusina rakenteina rekombinaation kautta. Tietoa voidaan käyttää lääketutkimuksessa, uusien luonnonyhdisteiden etsimisessä, genomilouhinnalla ja synteettisessä biologiassa, solutehtaiden rakentamisessa. Dear opponent, I now call upon you to present your critical comments on my dissertation.
So, dear colleague and co-authors, dear respondent and member of the audience, it's a pleasure for me to take the role of opponent for examination of your thesis work, Lassi. First of all, I want to thank the Professor Sivonen and you also for inviting me to debate about this wonderful project that, you know, I know a little bit because I have followed the development of your work during the 2 past years, because that was really interesting for me on a scientific point of view, because we are also interested in laxaficin synthesis. And so I have to say that in France, we are not used to such an examination of the thesis work with a single opponent, because in general, we are at least 3 to interact with the candidate, and I hope I will doing this in the right way. So among the compound we are extensively studying in the CREAB lab of the laxaficin family of compound, as you know. And this is certainly a reason you chose me as an opponent. I think we were first to report their total synthesis, and now we turn our attention to their ecological role and their environmental fate by examining their connection with an herbivorous rather than fed on cyanobacteria without being affected by their toxicity. So your thesis work is entitled Cyanobacteria Specialized Metabolites, Biosynthesis, Bioactivity and Structure. On a general point of view, what I have to say about the thesis dissertation is that the thesis is really well written and pleasant to read, and you provide also adequate citation. Nevertheless, I cannot understand Finnish, and so that was not so easy for me to follow the presentation, but I think you present it in a very didactic way, and you really focus on the main result you got, and for my impression, this remained very accessible for a large audience. So you clearly present the main goal, so the discovery of new metabolites through combined genome sequencing and genome mining approach. You isolate a lot of compounds during your thesis, and you analyze and characterize their structure very well through analytical chemistry method. And you also screen their bioactivity as antifungal compound that made so really a good overview on this compound. And I think through the method you implement, you highlight that the method you choose were really nice, and you show also that they have a broad scope of applicability. And this is also proved by the fact that you have produced 3 articles during your thesis. And when I go through the web literature, I have seen you recently publish also a review on the diversity of cyclic lipopeptide. So that is a point that has to be highlighted also. So you have used a wide set of methods encompassing cultivation of cyanobacteria, genetic methods, bioinformatics, chemical analysis, bioactivity assay. And this open a question for me. What is your main disciplinary field of expertise? You are more a microbiologist. You consider you as a chemist.
Yes. Okay. That's a good question. I think I see myself as a microbiologist, and I think that I would like to see the main results that I think I played the most biggest part myself is the DNA extraction and the analysis of the biosynthetic gene clusters. And
Okay.
maybe the chemistry part is maybe the one that I've been like more learning in this process. And
so this is why perhaps there is something I appreciate at the beginning of your discussion. You make a distinction between specialized metabolite and secondary metabolite. And I think the distinction you made is really crucial for the people reading such a literature, because as you said, secondary metabolite, that will be a kind of metabolite we don't have to care of. But in fact, using the term specialized metabolite, that give more focus on their that emphasize their potential ecological role. And this is really a point I have appreciated in your introduction. What seems to be for you the main point of interest in cyanobacteria is their ability to produce specialized metabolite for their use as therapeutics or potential drugs. And so I have a question concerning them. What made this metabolite so interesting for such an application? What will you say if you have to defend this point as to consider so secondary specialized metabolite from cyanobacteria as potential drug leads?
so you mean why do I think that the cyanobacterial specialized metabolites are good like drug lead source?
Yeah, that's right.
Yeah, I think that with the cyanobacteria there are many compounds that we don't know, and I think that that's a good starting point, because we have a lot of antibiotics and other drugs on the market that are well characterized, and with the organisms producing these common drugs, there is not that much new things to find. And with cyanobacteria, we constantly find new structures, and I think that's the main reason to focus on cyanobacteria.
So if I correctly understood, the main point will be the chemiodiversity found in cyanobacteria.
Yes.
And so we can be used for finding new drugs. Okay. And so, but if we look back to what is found in the literature, there are not so much drugs that are derived from cyanobacteria. And we are studying laxaficin for almost 30 years. And these peptides have not led until now to a template for drugs. So this is a little bit difficult. For you, what is the main problem to made laxaficin a potential drug? Laxaficin type peptides.
Yes, I think, I don't know, I don't think there are any clinical research on laxaficins. There are some of the bioactivities for these cancer cell lines and antifungal fungi that has been tested, but I think one of the issues is that we don't know the mechanism, how they work, and also that we need the synergistic effect, so we need two compounds, and this is, I think this might be problematic in the drug development. But yes, I don't know if I believe that laxaficins themselves will ever be a drug, but I think from the what we know about the structure and the biosynthesis, I hope that this can be applied elsewhere in drug development.
I fully agree with you concerning the fact that we have so this synergetical effect between laxaficin A type and laxaficin B type that made uneasy to understand how they work at the level of the cells. But might we have some idea of their toxicity in general? Are they specific for one class of cells, or are they fully unspecific? And that can be a clue concerning the way they act on cells. Did you think they really reach a special target, or what is your idea on how they affect the cells? So simply.
I think that there was some review from this fall where they listed some of these compounds, and they also mentioned laxaficins, and they were speculating that it's like a two-step process to attack the cell wall somehow, or the lipid layer. But I think it must be somehow quite specific, because with the scytocyclamides, at least, we also studied this with several fungi and several bacteria, but the only susceptible strain was the Aspergillus flavus. So it has to be quite specific, I think, the mechanism.
Yeah, but so as you point out right now, the specificity seems to be directed against to be an antifungal activity. But if we look to human cell lines, we cannot distinguish between cancer cell lines or normal cells, and this acts equally on both cells. So, and if we look at the level through microscopic technique, what is first the first effect we see is at the cell wall, and there are some blubbing at the surface of the cells. So it seems that the laxaficin interacts with the cell wall. So as you said, you were correct with this point. Among the different, so as you said, the cyanobacterial compound have not led to a drug, except for dolastatin 10. And this dolastatin was the development of development of dulastatin was really impaired by its toxicity. And so the researchers have tried to reduce it through SIR study. But as you said, it was not sufficient to turn dulastatin 10 itself as a drug. And then they do some modification that led to vedotin, but so you also write in your thesis that one of the main points to be considered is the Lipinski rule of 5, considering peptides. Do you think the Lipinski rules can be applied to peptides?
Yeah, I think I think in the conclusions of the thesis I write that I think that we need to start looking for these drugs also with non-traditional methods, and the Lipinski rule, I think it's maybe a bit outdated, but it's, or not outdated, but it's been used a lot, and you know, we should look for the chemical diversity maybe a bit also.
Yeah, I agree with you. The Lipinski rules do not really apply to the peptide. That's why we have to look out of this such kind of rule, and peptides open new opportunity. But if we look directly to the Lipinski rule, not one peptide will pass this filter, because there are plenty of hydrogen and acceptor bond and hydrogen donor bond. If we continue with dulastatin, I was surprised to in your thesis, because you highlight this drug because it's the sole one that was derived from a Cyanobacteria to reach the market, but I do not found the right term to define the vedotin. You know the right term or not? If you have to qualify vedotin, what is the name that is currently used to qualify this special type of drug?
You mean, what's the name of the drug?
Of the class of the drug, because the vedotin is a new class of drug. You don't remember? ADC, antibody drug conjugate.
Okay, yes.
This was, you have to mention it in your thesis work, or you should have to mention it, because that was the first ADC to be marketed, and also one of the first marine drugs to be marketed using this technology. So it's a kind of huge success from the Cyanobacteria research, etc., etc.
Yes. Yeah, I don't think, yes, I not mentioned this in the thesis. So.
Yeah, I'm sure you read it, because when you come through the publication, you have these terms that appears most of the time. Yeah. Okay. As you say, you define you as a microbiologist, more interesting in finding new or studying cyanobacterial genomes. And I have a very general question concerning the genomes of cyanobacteria. Are there a lot that are accessible through database, or this is not a phyla that is as much studied as the other bacteria? Or will cyanobacteria rank between all the bacteria in terms of genomic studies?
Yeah, I think with cyanobacteria there are not that many published genomes, and especially I think like in my papers we published 2 complete genomes, which I think there aren't that many complete genomes, but I think it's getting, there are now more there. It's increasing the number.
So because I was interested also by this question, and so this is also to point how new your thesis was, because from the whole of the bacterial genomes that are actually accessible, there are only 0.6% that concern cyanobacteria. So you really made some kind of advance doing your work, and so this is pleasant to have this information also that show that your work was really at the top concerning such question. So I will try to go much deeper between the different compounds you have studied, and so I will begin a little bit backwards or backward of your presentation, because I have to say that when I considered the 2 parts of your dissertation, I was really a little bit more uneasy considering the part dedicated to pseudo-spermidine and nodularin, and I have the impression that that was in fact some like a side project for you. That's a correct. It's my impression, correct or not? Because I have think reading this work that it was for you a kind of initiation to genome sequencing and to all the methods you will implement in the last steps of your thesis. That's right, or?
Yeah, partially it's right. Actually, the situation is that the pseudospumigine article, I was working on my master's thesis in the group, and from these results, the pseudospumigine article was published. And so after this, I continued with the PhD project, and we included this in the thesis.
Okay, so my impression was correct, and this was also we can see this because of the date of publication, of the time of publication. One was from 2017, and the other were 3 years or 4 years later. Yes. In the article concerning pseudospumigine, you explain that you need to have actinic strain before the DNA extraction. And did you perform this experiment, and have you faced any difficulties to have arsenic strain if I correctly remember, that was a not stock strain, no?
Mm.
Yeah.
The cultures, these Brazilian cultures, we got from we were collaborating with some Brazilian researchers, and they brought the cultures to Finland, and myself I haven't been working on purifying these cultures, and it was pretty much what it was.
So you have the chance, if I correctly understood, to have the cyanobacteria as an arsenic. Okay, because for what I have read, this is not so easy to get arsenic culture, and I wonder how this is performed.
Yes.
But so if you do not have done this work, so perhaps you don't know how it works and how many times you need to have this arsenic strain.
Yeah, with these cultures, I don't know, but also during my time in the group, I've been attending some sampling trips and also practicing the isolation of the cultures. And I think there's one culture that I was successful with purifying, at least to some degree. I'm not sure if they're arsenic, but it takes expertise and some time.
And so we have also tried to cultivation of cyanobacteria that are producing laxaticin in our lab, but that was marine species. And I have to confess, we were fully unsuccessful to cultivate this cyanobacteria. This is certainly a problem that is related to the fact that are marine species, and so this was really not so easy. Did you know that? Did you know? Yeah. Is there any obligation to have arsenic culture now with the advance of genomics to be able to study the genome of a bacteria, or we can do that in another way?
No, sorry, can you repeat?
Yeah. Normally, you need arsenic cultures to extract the DNA and to make the genome, the genomic studies. But as I said, for example, in our case, we were unable to have arsenic culture from a cyanobacteria. Might we hope to apply some genomic studies on this bacteria without having arsenic structure, culture?
I think actually 2 weeks ago, a colleague of mine also defended his thesis, and I think he mentioned about this, that actually some of the cultures that we consider arsenic in our culture collection, when we sent them to sequencing, sometimes we realized that they were not actually arsenic. But I think it might be possible to assemble the genome even with if there's something else. But okay, hey, I'm not.
Yeah, I think that through the advance of computer science and all the genomic stuff with NGS, so this is nowadays possible to assemble quite complicated genome and to do some metagenomics study. And so it seems that it's possible to learn about learn about mixture of cyanobacteria also. I don't know how, but it seems to be possible also. If I, yeah.
Usually the contaminants are not cyanobacteria, so you
can discard it.
Yeah, distinguish the genetic material.
So, but I'm not a specialist. I'm unfortunately only an organic chemist. So this is not my right field of expertise, but I read a lot about this. And so I think we have to consider this metagenomic approach as also an option. If I'm correct, the pseudospumigene are a trypsin inhibitor. Yeah. And for quantifying their activity, you use a peptide substrate containing a coumarin. Okay. But so you are during the quantification of the inhibition process, you observe variation of the fluorescence. That's correct also?
Well, I do observe, what did you say?
The fluorescence level will vary during substrate cleavage, no? You use a peptide with a coumarin.
Yes, yes.
Okay.
Yeah, the trypsin clears the coumarin and its fluorescence.
Okay, so my question is related to the substrate. At the beginning, when the coumarin remain connected to the peptide itself is fluorescent or not?
Not, no.
Not. So you have quenched substrate.
Well, yeah, I guess so.
Okay, and the fluorescence rise when the coumarin is cut from the peptide.
Yes, yes.
But what is in your substrate? Did you know what is the quencher of the coumarin fluorescence?
Oh, no.
Yeah,
I just can't do it. Yeah.
It's not a problem. That was just for my curiosity, because when I have looked to the peptide sequence, I could not catch it within the sequence of the peptide what was the quencher of the coumarin. And so that was a little troubling for me. I guess this is the arginine, and you have a kind of Pikachian interaction between the arginine side chains and the coumarin rings, and so that repress the fluorescence of coumarin. When you cut down the coumarin, then you have the coumarin that is free, and then you have the fluorescence. Always concerning pseudospumigene, you have a C-terminal acid, that amino acid that is an arginal. And so the arginal is characterized by an aldehyde function. And is this aldehyde function important for trypsin inhibition or not?
So is the arginine important for trypsin?
And more especially within the arginine, the aldehyde functionality.
Which functionality?
Aldehyde. COH.
I don't know. I think that with the trypsin inhibitors, usually they have this. The amino acid is something with this, I don't know, guanidine group with the amino, and this is more characteristic to the trypsin inhibitors. But I cannot tell. I think there are some spumigenes with arginine and arginинol, but I can't remember how this affects the potency.
If we consider the enzymatic reaction and the enzymatic catalytic site of trypsin, this is a serine protease. And so you have a serine that will be linked directly to the aldehyde. So this is really a key point in the activity of the pseudofungin. If you replace the aldehyde by a carboxylic functionality, then you will lose the activity, I guess. So, yeah, if you want to comment this, so it's just.
Yeah, I just think that there's also these trypsin inhibitors with the hydroxyl, but I'm not sure about this.
Well, you said at the beginning of our discussion that you are more a microbiologist, and so I'm asking questions that are more in the field of medicinal chemistry. That was just to have a look about that, but
yes.
You cannot answer all the question, and I prefer you said, okay, I cannot answer this question than giving some explanation that will be awkward or something like this. Concerning the nodularine, I have not so much question. If I'm correct, you see that the production of this compound was twice higher than in other cyanobacteria. Is that correct?
It's about the same concentration that is seen in some nodularia strains, Baltic Sea, but it's there are some nostocs that have been described with nodularian, but it's been very low amounts. So this is I think first strain that is not a nodularia that has this kind of production concentrations.
Okay, that was an information I have not catch. Just a remark concerning the nodularine. In the article, you have a figure that describes the nodularine structure, but so I was uncomfortable with this figure because so this is presented in a not so correct way, and but so I know you will not be able to modify it right now, but the L2, L4, L3 would have been if possible, to be modified, and by indicating that L2 or L4 are CH3, L3 is an H4, PSPA, instead of giving the name of the whole amino acid, because this is not so the structure is not so correct by giving
in the article.
Yeah, in the article, but it's too late and it's not so of great importance. So we will turn on laxaficin, because this is the main part of your work. And I have to say that I have tried in 2017 to mine the same strain as you for the cytocyclamide, but unfortunately I have not all the competence to do that properly, and I have tried to interest some colleague in the mining of the PCC, which I don't remember. The one, the cytonema of Manii, and the colleague were overbooked and say, okay, try to do that by yourself. And as I have no the competence, I think we missed the opportunity to discover the BCG you have highlight in your thesis.
Okay.
So congratulations for this.
Thanks.
And so it is not so common to finish a thesis having given its name to a novel class of compound. If I'm correct, the enamide are derived from your last name, no?
Yes.
Yeah.
Yes.
But so you point out also in your dissertation that we are facing chemists, microbiologists, a lot of difficulties to found the compound because of those type of naming. Do you think there is any possibility to have a kind of systematization of the family names or of the name of the cyanobacterial products? Did you know if someone has been interested in this?
I think maybe with naming enamides as they are, maybe we were not really like consistent in this approach to have more systematic naming, but I think with the laxaficins there have been publications. I think maybe your group has also made these publications with naming the variants by telling in the like something valley in laxaficin A to try to highlight the difference. But when you have multiple variations, then the names will be just huge, and then it won't serve any purpose anymore if you have like 4 or 5 or more variations and you want to tell each of them in the name of the compound. So I don't know if there's a good way to do this, or what will be the best way.
This remains really a problem, because I think we lose some information, and sometimes you pass and you didn't look to some products that are related to the laxaficin compound, even if they exist. That was for me the case when I look through the literature, and I found the cytocyclamide first mentioned in the thesis of Mr. Groove. You said it in your work, but that was not so easy to find the information. That was not navigable information and so easy to find. And with such a naming, without any systematization, that makes the thing very difficult, I think. Perhaps some people will find a way to systematize the naming of compound issuing from cyanobacteria. This remains an open debate, I think. A point that is unclear for me concerning laxaficin A derivatives is related to their location in the cyanobacteria. Are they extracted from the cyanobacteria to form a cloak at their surface, or they remain within the cyanobacteria? Did you have a look to this, or?
We only did extracts of the freeze-dried cell mass. So at least I don't know if they're excreted. Usually, I think the starting point is that we usually think that they are in the cell, and we focus on that, and we find the NRPS products in the cell. But did you say that it could be like on the surface of the cell, the compound?
Yeah, because this is also in connection with the ecological role. There is two ways to see this, whether they are at the surface and they act as repellent, or they are inside and they act as repellent because the grazer will feed on it and so feel uneasy to consume them, or they are repelled because of the smell of the laxaficin. This is unclear for us. So that's why I was asking this. But if you have not the answer,
yeah, no.
the question remains open. And in direct connection with this, if we consider that the laxaficin remain inside the cells, how will the cell regulate the level of laxaficin? Because we might also, if the laxaficin act on membranes, we might expect that there are toxic also for the bacteria. So is there any mechanism for the regulation of the laxaficin production to avoid self-poisoning?
Yeah, I think that, yeah, I have not thought about that so much, but I think that most of the bacteria that have been studied with laxaficins are not the producers, but the bioactivity. I think usually the bacteria in general are more resistant to laxaficins than eukaryotes, and also I think the synergistic effect has not been reported on bacteria, but it's strong on eukaryotes. So
no, let's see. it's just directly in connection with the cyanobacteria, how the cyanobacteria itself regulate its own production of laxaficin, because as the laxaficin are shown to be toxic, and even more when you combine the two, laxaficin A and laxaficin B, if they remain within the within the cyanobacteria, I think we might expect also a kind of toxicity, because the level will be so high that the cyanobacteria will experience a kind of self-poisoning. So that's why I was asking if there is a mechanism of auto-regulation of the laxaficin production. If you have looked at the genome, if you can find a kind of genes that will provide, that will code for a peptidase that will be specific for one of the laxaficin and will cleave the laxaficin ring in order to repress the toxicity of the laxaficin by opening of the peptide ring.
Yeah, I don't know how it's regulated.
Okay.
So answer the question, but yes, I still doubt that maybe the product is not toxic to the producer, or not as toxic.
Yeah, why not? But I say this because I don't know if you have read the article on different antimicrobial that was published in 2018. And that was, I think, a Chinese group that was collaborating with someone in Britain. And they, when they looked to the production of daptomycin, there was a repressor genes that code for peptidase that was itself involved in the degradation of the daptomycin. And that was a dipeptidase. That's why I was asking this, because what we suspect is that the dipeptidase that is able to cleave the laxaficin B, we reported recently, might be in fact from the cyanobacteria.
Oh, okay. Yeah, I think it will be maybe, or it will be interesting to look for these peptidase genes in the genome, but yeah, we have not done this. And
okay, that was the point. To if you might have time to look, if you can found any so peptidase, dipeptidase in the genome you have, this will be for us of great help. Yeah, one point that is also directly in connection with the genome and the laxaficin A structure. There are two notable exceptions in the laxaficin A family: the homotamine A, which had DHB instead of the EDHB, and the trichomamide A with the L-serine instead of the EDHB. And now that you have deciphered the biosynthetic gene cluster, what do you think about this laxaficin A variant?
you mean hormotamine?
Homotamine A. Homotamine A, you have a ZDHB instead of the EDHB. And for trichomamide A, you have a L-serine instead of the EDHB. And when I look to your results, I cannot catch how it is possible. And could we suspect there is a false interpretation of NMR data, or because, so when I look to the trichomamide A, we have not tried. We synthesize it, but the NMR information do not match the one found for the natural product. So we are suspecting that there is a false interpretation of NMR data in the case of the natural compound. And so because of the biosynthetic pathway, we have to expect that for each of the compound at the position of the EDHB, we have a EDHB, no? For the laxaficin A. I don't remember what is the numbering of the residue, but
yeah, it might be just that it's a different domain that's not dehydrating the serine. But what did you mean? Did you mean that the serine is misanalysis and it should be the DHB in the trichomamide?
Yeah, I would think so. Or the E-serine, I don't understand this result because there is a kind of consistency between all the laxaficin A variant, and they all have at this position a EDHB, except in one case with this serine. And I will understood to find a threonine at this position because the threonine is the precursor for the EDHB, but L-serine, this made me a confusion about this.
Yeah, could be that there's some mutation in the adenylation domain, and it, yeah, maybe, yeah, I don't know where this comes from, but if it was the threonine, maybe the condensation domain still could do the dehydration. I don't know.
Okay. But because there is another example, this is in the laxaficin B family. When we look to the table, the table 2 you report in your thesis work, if we look to the lobocyclamide, you have a DGLN instead of the LGLN, so glutamine, deglutamine instead of L-glutamine, and because of the consistency or at this position, might we think that there is something that is falsely reported in the case of lobocyclamide, and that in fact we really have also a conservation of the LGLN position that's remain relatively stable with throughout all the laxaficin variant. Is that
it would be interesting to sequence the trichomamide producer to see what the cluster looks like.
okay?
Yeah, that's what I can say. Because yeah, just yeah, it's very exceptional that it's only one variant with the serine and not the DHB. So or some systematic like study on how it affects the bioactivity, if it's serine or
I don't remember if they have tried to combine so the trichomamide C and A to look if they got some synergetic effect, as in your case. So another interesting feature of the laxaficin is the alternation of the S and R stereochemistry. Do you think this has an impact at the structural level for the laxaficin? Do we have any idea on how the laxaficin ring is structured, or at a 3D level? So,
well, at least we have not paid attention to the 3D structure or the how it's organized. So I don't remember seeing any reports on that.
For my side also, I haven't found any report on this, but it seems that from other literature sources, that when you have a ring with alternation of S and R stereochemistry, the ring will have a kind of flat structure that allow to have molecular assembly and to form some kind of tube. And another point of interest is that doing this, you have all the chains that will be on the face of the ring, and all the OH from the alcohol functionality will be on the same side, and that can induce some chelation phenomenon with metal, so such as calcium. Have you looked to the possibility of laxaficin to chelate any metals, or this was out of focus of your study?
This is the first time I hear about such.
You heard about this?
But yeah, I don't know. Sounds interesting, but yeah, no, we have not thought about
it. Yeah, so I have plenty of questions, but I will try to reduce. Said me if so the custos, if I'm too long, I have 2 or 3 more questions concerning the FAL enzyme that begins the synthesis of laxaficin in general. You say that hexanoic acid is the first to enter in the sequence, so in the process. But could it be possible that it will be for enamides the 2-hydroxyhexanoic acid that directly enter, that is also capped by the FAL enzyme? In other words, how you are shown that is hexanoic acid, the first precursor of the fatty acid?
And not the hydroxylated one.
Yeah, and not 2-hydroxyhexanoic acid.
Yeah, I can't, I can't understand. I don't think I have an answer for that right now.
But if so, on other words, how can we try to verify if this hypothesis is right or false? Is there any possibility, for example, to feed the cyanobacteria with hydroxyhexanoic acid as you do for the proline derivatives, and to see if we have an higher expression of?
Yeah, well, you could look into it like with labeled fatty acids or something to see which it is, but I believe we had, or I had a reason to say that it's the fatty acid and that the mechanism works on that, but yeah.
Okay. So you are also interested in the 3-hydroxy-4-methylproline production, and you did a feeding experiment with 3 of the isomers of the 4-methylproline, but why not the 4 stereoisomers?
Why not? I think you mean why we only use 3 of the isomers?
Yes, but this is because one of the isomer was not commercially available, or?
Yeah, I think we tried to have all the isomers, but it was too expensive, or it was not available. Because we would have done all the isomers if we could have.
And so I can we all face some financing problem when we did research, and we have to find some solution to spare money. And but at this level, if I'm correct, all the 3 stereoisomers are equally incorporated in the NMate sequence. Or so there is no stereospecificity of the adenylation domains. That's right? Or there are some stereochemical chemical preferences?
The level is pretty much the same. The difference is not very big. So
okay.
I would not draw any
any conclusion about that.
Yeah.
That was an impression I have also, but it was not so easy for me to catch the information, because we are expecting some level of stereospecificity at the adenylation of the adenylation domains, but in this case, this seems to not be the case. Concerning the biosynthesis of the 3-hydroxy-4-methylproline, you hypothesized that that this compound was obtained starting from leucine. Yeah. And I have a question of certainly of an organic chemist, because I don't know how it works at the level of the cultivation of the cyanobacteria. If you introduce leucine that will be enriched in nitrogen 15, could we expect to have to observe the insertion of 15 nitrogen 4-methyl 4-methylproline with a 15 N nitrogen mark, or to this will be to confirm the production of the 4-methylproline starting from leucine.
Yes,
Ah,
we could, but yeah, I think yeah, what we did that we were feeding the leucine and just analyzing the levels, what's incorporated, and there was no difference. But I think with labeled like intermediates, you could see how the biosynthesis works. If it's incorporated to the product.
Okay. Yeah. I look a little bit about the question I have. What was unexpected for me was the fact that the hydroxylation of the D-leucine to turn them into 3-hydroxy-D-leucine was performed by tailoring enzyme, and this was not included directly in the NRPS assembly line. And so you discussed that very well in your dissertation and through your article. I have just to remark, you attribute to us the fact that cytochrome P450 might be a possible actor in the beta hydroxylation. In fact, this is an hypothesis that we found in an article authored by Max G. Kreil in amino acid peptides and proteins in 2013, and this was not our hypothesis.
Okay.
Okay, this is just to correct this, because there's some other guys that do this observation, and they observed this in the case of scilamamicin. This is a peptide that contains also nitroxylated leucine. What was really surprising for me in the case of enamide is the presence of the carbomoylated homoserine. How did you identify the presence of this residue within the enamide? Is that only using the mass spectroscopy, or the combination of the mass spectroscopy and the NMR?
Definitely also the NMR, but yes, I didn't do this analysis myself, so
you cannot.
But it was based on NMR.
Yeah, okay. Because when I read first your article, I was intrigued by the fact that you have also the 3-hydroxyglutamine that will be isobaric to the 3, so the carbomoylated homoserine. But in fact, when I look to the MS spectra and the NMR, there is no trouble at this level. This is with certainty an homoserine that you have in this position, because all the data give this point. Okay with it. For determination of the structure, you also use Morphy derivatization. And I have a question concerning this carbomoylated homoserine. Did it stand the Morphy condition of, because you start first with an hydrolysis of the peptide, and then you derive all the amino acids. That's right? and the carbamoyl moiety remain before the Marfé derivatization or not?
Yeah, I'm not sure about that. Yeah, yeah, I'm not sure.
Yeah, because so that was an interesting point for me, because whether you found it as an homoserine or you found a derivative in the Marfé analysis that was without any how to say amino acid standard. And so that's okay, but if you have not the answer, we will not spend a lot more time about that. Yeah, because just to conclude about this point, this can be also another way to characterize the presence of a carbomoylated moiety in a peptide, is to perform a controlled hydrolysis of the carbomoylated moiety through, for example, saponification. I have found this in the case of crocagines. That was something that was published in Angewandte Chemie in 2017. And they also identify within this article some carbamoyl transferase that are applied to homoserine.
Okay. Yeah, because yeah, it was quite hard to, or we didn't find other structures with this carbamoylated homoserine, but okay, so they exist. So
yeah, that was so unexpected when I read your article. That's I performed some literature studies also to understand where it can come from and how stable is it, because carbomoylated moiety have not a reputation of so high stability. And so that's why I looked to this, because that was really intriguing. So the respondent Cursus, member of the audience, I have to say that considering the quality and the amount of work that you have produced during your doctoral project, I was really impressed. And you tackle also not answered aspect of the biosynthesis of laxaficin. You provide new information and in the processing of the amino acid entering in the laxaficin synthesis. And so it is my pleasure to propose acceptance of your dissertation to the faculty, because this is really nice and well done work.
okay, so hey, thank you very much, Nicolas, on the discussion. It was much more convenient than I expected. And if anyone present wishes to make any comments concerning my dissertation, please ask the Kustos for the floor.
There is no questions. This public examination is completed. Väitöstilaisuus on päättynyt.
Okay, so thanks everyone for coming. Kiitos kaikille. Yeah, there's coffee and cake outside the room shared for us. Thank you.
Professor, our service will contact you soon for the grading. So thank you very much.
You're welcome. It was really a pleasure.